Hello, my name is Stephanie Baldwin and I'm currently studying a third year of Bachelor of Biomedical Science at the University of Otago. Over the summer, I've been completing a summer research project with the Physiology department and supervised by Associate Professor Pete Jones. I would like to express my sincerest gratitude to Dr. Ailsa Goulding and the Otago Medical Research Foundation for funding my scholarship. It has been an invaluable opportunity that has allowed me to combine my passions for human physiology and also helping people. Overall, my project focuses on the investigation towards a new possible drug target that could be used to treat the symptoms of cardiovascular diseases. A normal heart contraction is a controlled process that occurs in response to an external stimulus on the tissue. This maintains a steady heartbeat rhythm. However, in pathological cases, this regulated process is disrupted and the contractions occur without any external stimulation. This causes an abnormal heart rhythm known as the state of arrhythmia. Within heart cells, there is a calcium store known as the sarcoplasmic reticulum, which releases calcium when stimulated in order to activate a cardiac contraction. The ryanodine receptor lies on the surface of this store and has a threshold that can control the release of calcium. An enzyme known as casein kinase has been recently discovered to have the potential to change this threshold, possibly altering the occurrence of arrhythmia. During a normal stress response, multiple cardiac proteins pump more calcium back into these stores. In a normal cell, even with the added calcium, the threshold is not reached, so the calcium does not get released from the store. Through changing the activity of casein kinase, it is thought to sensitize the ryanodine receptor, which would reduce the threshold for calcium storage. In these cases, with a normal stress response, the increased level of calcium passed the lowered threshold, releasing calcium into the cell, which activates an inappropriate contraction. This process relates to the analogy of holes in a water bottle, the bottle being the calcium store, the hole acting as the threshold, and the water representing the calcium. The first bottle illustrates a normal cell's threshold which remains higher than the water level, so it remains in the bottle. On the other hand, the second bottle represents a reduced casein kinase activity with a lower hole, meaning the water level reaches threshold easier and escapes the bottle. The study tests this hypothesis by observing isolated heart cells under various pharmacological inhibitors and by altering the enzyme's activity through genetic alteration. The occurrence of calcium release in the cells when placed under varying external conditions is detected as wave formations using a microscope.